DNA before today.
DNA was discovered in 1869 by a German biochemist named Friedrich Muster.
But in 1953, James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin discovered the structure of DNA, a double helix structure.
They also realized that the molecule carries biological information. This led more scientists to research more information about DNA.
Francis Crick was born in 1916. He discovered concept 19, where the DNA molecule is shaped like a twisted ladder, and contributed more to discovering the DNA structure.
James Watson was born in 1928, and he was considered the father of DNA. He also contributed to discovering the DNA structure by discovering DNA itself and the double helix molecular structure.
Crick and Watson discovered the chemical structure of DNA in 1953. DNA structure is considered to be like a twisted ladder, otherwise known as the double helix structure.
If you think of the double helix structure as a ladder, the phosphate and sugar molecules would be on the sides, while the bases would be on the rungs. The bases on one strand pair with the bases on another strand.
Putting these together in a strand will form a DNA molecule that contains genetic information.
Alfred Hershey was born in 1908. He discovered that DNA, not protein, was the genetic material.
Martha Chase was born in 1927. She proved that DNA carried genetic information alongside Hershey. They also conducted the blender experiment, where the scientists discovered that protein did not have genetic material.
DNA is made up of molecules called nucleotides. Each nucleotide includes a phosphate group, a sugar group, and a nitrogen base. There are 4 types of nitrogen bases, which are adenine, thymine, guanine, and cytosine. Nucleotides are attached to each other to form 2 strands that spiral and create a double helix structure.
DNA molecules are so long that they can't fit into cells without the correct packaging. Therefore, they are coiled tightly to form chromosomes. Concept 19.
Before Watson and Crick solved the DNA structure, there was already existing information of DNA and what it was made up of due to X-ray crystallography, but what they couldn't quite solve was the structure of the molecule itself.
X-ray crystallography provided the final clue that answered their question. They finally figured out what the structure was, which was the double helix.
They showed alternating deoxyribose and phosphate molecules that formed the twisted ladder. The rungs of the ladder were formed by complementary pairs of nitrogen bases, where adenine always paired with thymine and guanine always paired with cytosine.
The blender experiment.
In 1952, the blender experiment was conducted by Martha Chase and Alfred Hershey. They both conducted the blender experiment, where they used a wearing blender to violently disturb the infected bacteria, which caused the protein shells to detach from their host.
The test was performed twice with E. coli.
They conducted this experiment in order to show that proteins carry genetic information.
The result of the test were that the radioactive sulfur was found in the supernatant. The second result, radioactive phosphorus, was found mostly in the cell fraction, where a new generation of infective phage may be alone.
In conclusion, the active component of the bacteriophage that channeled the infected characteristics was the DNA. There is a relation between DNA and genetic correlation. Thank you for watching.